Potency, unraveling the differences in drug evaluation. In the world of pharmaceuticals, ensuring drug safety and effectiveness is paramount. Two critical concepts in this realm are assay and potency. While often used interchangeably, these terms hold distinct meanings crucial for understanding drug action. This essay delves into the differences between assay and potency, highlighting their significance in drug development and patient care. Understanding assays, measuring biological activity. An assay is a test. It measures a drug's biological activity. It assesses how a drug interacts with its target to produce a response. Assays are crucial in drug discovery, revealing a drug's mechanism and potential benefits. Various assays measure specific drug activities like binding, enzyme, and cell-based assays. Binding assays determine how well a drug binds to its target. Enzyme assays measure a drug's effect on enzyme activity. Cell-based assays evaluate a drug's effects on living cells. The choice of assay depends on the drug and response being studied. Assays provide valuable data, allowing comparison of drug activities. Exploring potency, the strength of a drug. Potency refers to the strength of a drug. It's a measure of how much is needed to produce a specific effect. A highly potent drug works at lower doses, while a less potent drug needs higher doses. Factors influencing potency include affinity and intrinsic activity. High affinity means strong binding, increasing potency. High intrinsic activity means a strong response. Understanding potency helps determine the right dose for patients. Too potent can be toxic. Not potent enough can be ineffective. Distinguishing assay from potency, why it matters. While assay and potency are distinct concepts, they are interconnected. An assay can be used to determine the potency of a drug. For example, a cell-based assay can measure the concentration of a drug required to inhibit cell growth by 50%, known as IC50. This IC50 value serves as a measure of the drug's potency. The distinction between assay and potency is crucial because it underscores the multifaceted nature of drug evaluation. It's not enough to know that a drug produces a particular biological effect. We need to understand how effectively it does so and at what concentration. Confusing these terms can lead to misinterpretations of drug data and potentially compromise patient safety. For instance, assuming a drug with high biological activity in an assay automatically translates to high potency can be misleading. The drug may have low potency due to factors like poor absorption or rapid metabolism, requiring higher doses to achieve the desired effect. The crucial role of assay and potency in drug development. Assay and potency are integral throughout the drug development process, from discovery to clinical trials and beyond. During drug discovery, assays are used to screen thousands of compounds for potential therapeutic activity. Compounds showing promising activity in assays are then further evaluated for their potency and other pharmacological properties. In preclinical development, before human testing, drugs undergo rigorous preclinical testing in cells and animals. Assays are employed to evaluate drug safety, efficacy, and potency in these models. In clinical trials, assays are used to monitor drug levels in patients and assess their response to treatment. Potency data is crucial for determining appropriate drug dosages for different patient populations. Once a drug is approved for market, assays ensure the quality and consistency of each batch produced. Manufacturers must demonstrate that each batch meets specific potency standards to guarantee drug safety and effectiveness. Ensuring drug safety and efficacy. The significance of understanding assay and potency. Understanding the difference between assay and potency is not just an academic exercise. It's fundamental for ensuring drug safety and efficacy. By accurately measuring a drug's biological activity and strength, scientists and clinicians can develop effective drugs. Understanding how a drug interacts with its target and its potency allows for the development of drugs that are both effective and safe. Potency data is essential for establishing safe and effective drug dosages for different patient populations, considering factors like age, weight, and overall health. Assays ensure that each batch of a drug produced meets the required potency standards, preventing the distribution of substandard or potentially harmful medications. In conclusion, while often used interchangeably, assay and potency are distinct concepts with significant implications for drug development and patient care. Assays provide insights into a drug's biological activity, while potency reflects its strength. Understanding these differences is paramount for interpreting drug data accurately, developing safe and effective medications, and ultimately ensuring patient well-being.